Welcome to the part 2 of our topic on the sources of drugs. For the outline of this presentation, the first part of our topic will focus on the continuation of the active principles of medicinal plants. We also have the synthetic drug sources, the semi-synthetic drug sources, the gene therapy, and the biopharmaceuticals. For the learning outcomes, this presentation aims to identify and describe the different drug sources and identify some examples of these drug sources. Let's start with the continuation of the active principles of medicinal plants. The active principles or medicinal principles of plants are substances found in plants that exert a physiological or nutritive function in the body. They are purified components of a plant which have studied and scientifically proven therapeutic functions. The medicinal value of plants or the crude preparations of medicinal plants is due to the presence of a variety of pharmacologically active principles such as alkaloids, glycosides, resins, oleoresins, gums, saponins, and tannins. Alkaloids are a class of basic naturally occurring organic compounds that contain at least one nitrogen atom. They are also defined as basic nitrogenous substances that is insoluble in water, less soluble in alcohol, and soluble in ether, chloroform, and oils. The alkaloids consisting of oxygen are solids. Examples of this are atropine, resarpine, emethine, morphine, strychnine, and quinine. The alkaloids without oxygen are liquids. Examples of this are nicotine, Pylocarpine, and Lobiline. The important alkaloids, along with their source and major pharmacological actions, are shown in the slides to follow. The first is atropine. The source of atropine is the plant Atropa belladonna, and its pharmacological action is cholinergic and muscarinic blocker. Cholinergic drug, any of the various drugs that inhibit, enhance, or mimic the action of the neurotransmitter acetylcholine, the primary transmitter of nerve impulses within the parasympathetic nervous system. The next is arculene. Arculene is an alkaloid obtained from the betel nut, Areca catechu, fruit of a palm tree. It is considered to be an agonist at both the muscarinic and the nicotinic acetylcholine receptors. It is used in the form of various salts such as a ganglionic stimulant, a parasympathomimetic, and an anthelmintic, especially in veterinary practice. The next alkaloid is caffeine. Caffeine comes from the plant Coffea arabica, and its pharmacological action is cortical and cardiovascular stimulant. The next is cocaine. Cocaine is derived from the plant Erythroxylum coca plant, and cocaine has been used since ancient times, and its excitotic and psychostimulant effects have been clinically recognized for over 100 years. Aside from this, it is now used as a local anesthetic, and it is also used for CNS excitation. The next is colchicine. Colchicine is derived from the plant Colchicum autumnale, and it is used as an anti-inflammatory for gouty arthritis. Colchicine is used to prevent gout attacks, such as sudden severe pain in one or more joints caused by abnormally high levels of a substance called uric acid in the blood in adults, particularly in humans. The next is emethine or cephaline. This alkaloid is derived from the plant Psychotria epicaquanha, and it is mainly used for reflex vomition, especially during GI irritation. Cephaline is an alkaloid that is induces vomiting by stimulating the stomach lining and is found in commercial products such as syrup of Epicac. Chemically, it is closely related to emethine. Emethine is a drug used as both antiprotozoal and to induce vomiting. It is produced from the Epicac root and it takes its name from its emetic properties. We also have ephedrine. Ephedrine comes from the plant Ephedra vulgaris, and it is mainly used as an indirect acting sympathomimetic drug. Ephedrine is a synthetic non-catecholamine agonist at alpha, beta-1, and beta-2 receptors with both direct and indirect actions. Ephedrine is a prescription medicine used to treat the symptoms of low blood pressure during anesthesia or hypotension. The next is epinephrine. Epinephrine, also called as adrenaline, is a hormone that is secreted mainly by the medulla of the adrenal glands, and it functions primarily to increase the cardiac output and to raise the glucose levels in the blood. Epinephrine is typically is released during acute stress, and its stimulatory effects fortify and prepare an individual for either fight or flight. So epinephrine is a sympathomimetic and a cardiac stimulant. The next alkaloid is ergometrine. Ergometrine is an ergot-derived oxytocic agent frequently used on obstetrics. It is derived from the plant Claviceps purpurea. It is an oxytocic and an alpha adrenoreceptor blocker. Its commercial name, methyl ergometrine maleate injection, is used to stimulate uterus and hasten parturition. After the parturition, this injection is useful in the treatment of retention of placenta and to induce the involution of the uterus. It is used in veterinary medicine in the control of postpartum uterine hemorrhage, removal of fluid from atonic uteri, to prevent prolapse uteri, and judiciously in terms of timing to aid in suturing the uterus after cesarean section or in replacing an inverted uterus. Another alkaloid is leptadine. Leptadine is derived from the plant Leptadenia reticulata, and this is a galactagogue. Its commercial preparation in veterinary medicine is Leptadine. This drug is indicated for deficiency, irregularity, or absence of lactation in large and small animals to increase milk yield. According to the manufacturer, it is also indicated for retained placenta. It is said to induce safe, gentle, and smooth expulsion of retained placenta and its threads. Another alkaloid is lobiline. Lobiline is derived from the plant Lobelia inflata or Indian tobacco. It is both an agonist and an antagonist at nicotinic receptors, although it is not structurally related to nicotine. Another is pilocarpine. Pilocarpine is derived from the plant Pilocarpus jaborandi, and it is a muscarinic cholinergic agonist used on the eye to treat elevated intraocular pressure, various types of glaucoma, and to induce miosis. When we say miosis, that is an excessive constriction of the pupil of the eye. Another important alkaloid is quinine. Quinine is derived from Cinchona officinalis. Cinchona officinalis is a medicinal plant, one of several Cinchona species, used for the production of quinine, which is an anti-fever agent. It is especially used in the prevention and treatment of malaria. Another is Resarpine. Resarpine is derived from the Indian snake root Rauwolfia serpentina. It is mainly an antihypertensive agent. Rauwolfia serpentina is a safe and effective treatment for hypertension. The plant was used by many physicians throughout India in the 1940s and then was used throughout the world in the 1950s, including the US and Canada. Another alkaloid is the Somneferin. Somneferin is derived from Withania somnifera. It is considered to be an immunostimulant and an anti-stress agent. The plant Withania somnifera is a winter cherry tropical to the Solanaceae family, and it is also used in treating a wide variety of illnesses, including asthma, diabetes, hypertension, stress, arthritic diseases, and also cancer. Another alkaloid is strychnine. Strychnine is an alkaloid found in Strychnus nux vomica. It causes excitation of all parts of the central nervous system with a characteristic motor pattern. Strychnine is a competitive antagonist at inhibitory neurotransmitter glycine receptors in the spinal cord, brain stem, and higher centers. Today, strychnine is used primarily as a pesticide, particularly to kill rats. Its use is restricted by law. Another medicinal principle or plant principle is the glycosides. The glycosides are compounds containing sugar or glycone and non-sugar aglycone parts joined together through an ether-like linkage. The pharmacological action is in the aglycone or the genin part, and the glycone or the sugar part determines its solubility, tissue permeability, and duration of action of the aglycone. On acid or alkali and enzyme hydrolysis, the glycoside breaks into two parts. So we have here the chemical structure of a glycoside, particularly a cardiac glycoside, and it shows that it has mainly two parts. Now we have the sugar part or the glycone and the non-sugar part or the aglycone part or the aglycone moiety. Our first example is the digitoxin and digoxin. These glycosides are derived from the leaves of Digitalis lanata and the Digitalis purpurea, and they are considered to be cardiotonic. So this plant here is the Digitalis lanata or the Grisean foxglove, and this one here is the Digitalis purpurea or the common foxglove. So they are said to be cardiotonic. When we say cardiotonic, these are drugs used to increase the efficiency and improve the contraction of the heart muscle, which leads to improved blood flow to all tissues of the body. Digoxin is used in both human and veterinary medicine to treat a variety of cardiac disease states, including CHF or congestive heart failure and atrial fibrillation. Cats are relatively more sensitive to digoxin toxicity than dogs. Another glycoside is the Stropantidine. Stropantidine is a cardiac glycoside derived from the seeds of Strophanthus combe. It is also considered to be a cardiotonic. Stropantidine is a cardiac glycoside which mechanism of action is similar to Digitalis, ouabain, and digitoxin. Another glycoside is the Nereuside. Nereuside is derived from the leaves and the seeds of the plant Nerium Oleander, and it is mainly a cardiotoxic agent or a cardiotoxic glycoside. When we say cardiotoxic, that is a substance that induces damage to the heart muscle. Another is the Cerberine. Cerberine is derived from the seeds of the yellow oleander, Cerbera tevetea, and it is said to be cardiotoxic and extremely toxic at higher doses. Although it is cardiotoxic, it has also some medicinal properties according to some studies. The yellow oleander seed, particularly its alcoholic extract, have been found to have anti-cancer property and antimicrobial properties. Another is the aloin. Aloin is derived from the leaves and the stem of aloe vera, and it is a glycoside with a wound healing property as well as antibacterial property. We also have the Mangiferin. Mangiferin is derived from the fruit and leaves of the mango tree, Mangifera indica. It has hepatoprotective and antioxidant property. The Mangiferin is a xanthone present in significant levels in higher plants and in different parts of the mango fruit, such as the peel, the stalks, the leaves, the barks, the kernel, and the stone. It is a promising antioxidant with tremendous health-related properties such as antiviral, anti-cancer, anti-diabetic, antioxidative, anti-aging, immunomodulatory, hepatoprotective, and analgesic effects. Another is the Stevoia Marine. Stevoia Marine is derived from the stem and the leaves of Stevoia cirrata. This glycoside is said to be cardiotonic and hepatotonic. When we say hepatotonic, that is a substance that is considered to be a liver tonic. A liver tonic is a substance that is tonic to the liver, meaning it is employed to normalize the liver enzymes and function. The Stevoia Marine, aside from its cardiotonic and hepatotonic property, has been reported to cure many diseases such as diabetes, atherosclerosis, hypertension, arthritis, malaria, and abdominal ulcers. Another classification of drugs are those drugs that are derived from synthetic sources. Synthetic drugs are generally defined as those drugs that are prepared by chemical synthesis in pharmaceutical laboratories. They can also be defined as a drug that is created artificially in imitation of naturally occurring substances to exert a specific pharmacologic effect. Examples of synthetic drugs are antipyretics, barbiturates, non-steroidal anti-inflammatory drugs, tranquilizers, anesthetics, and others. We also have the semi-synthetic drug sources. When the nucleus of a drug obtained from a natural source is retained, but the chemical structure is altered or changed, we call it a semi-synthetic drug. Semisynthetic therapeutic agents are a hybrid of natural and synthetic sources. Semisynthetic therapeutic agents are generally produced by transforming starting materials from natural sources into final products via chemical reactions. Examples of semisynthetic medicines are paclitaxel. Paclitaxel is an anti-cancer drug derived from the Pacific yew tree, Taxus brevifolia. We also have the derivatives of an antagonist of morphine. Another are the dihydrostreptomycin from streptomycin, and the semisynthetic penicillin from natural penicillin. We also have the gene therapy. So gene therapy is also another drug source. Gene therapy means the prevention or treatment of diseases through manipulation of a gene function. It is the insertion of a specific genes, therapeutic genes, exogenously into animal cells. The concept of gene therapy has its origin from the fact that manipulation of gene expression could change the function of abnormal cells as manifestation of an existing gene and thus the course of a disease. Recombinant DNA technology forms the basis of synthesis of therapeutic genes. So we have here an overview of the types of gene therapy techniques used in the treatment of HCC. HCC stands for hepatocellular carcinoma, which is the 5th most common cancer and the 2nd leading cause of cancer-related deaths worldwide. Gene delivery into the host involves inserting therapeutic genes first into a vector. The vector can be viral vector. The example of a viral vector are retro or adenoviruses, or non-viral vectors. Example of non-viral vectors are liposomes, microspheres, and plasmid DNA. A vector with a gene is then introduced into the patient through either in vivo or ex vivo means. So this diagram here shows the two types of gene therapy techniques. The two types wherein the therapeutic gene will be introduced into the patient. The first type is the in vivo gene therapy, and the second type is the ex vivo gene therapy. The in vivo means involves injecting the suspension of the vector having the therapeutic gene intravenously or into the target tissue organ. So we have here the in vivo gene therapy wherein we are going to introduce a therapeutic gene IV into the target tissue or the organ of the patient. And this can be done through non-viral delivery system and through viral delivery system. So the viral delivery system involves the use of a virus. The virus could either be a retrovirus or adenovirus, and this particular virus contains the gene of interest, particularly the therapeutic gene. We also have the non-viral delivery system. In this case, we use a nanoparticle, and this will be used to introduce the therapeutic gene into the target tissue or organ of the patient. We also have the ex vivo means, the ex vivo gene therapy. The ex vivo method involves first inserting the therapeutic gene into stem cells obtained from the patient. This could either be blood or bone marrow, and inject the same into the patient. So we have here a stem cell that is isolated from the patient. Next, the cells will be modified in vitro, and the modified cells will be injected back into the patient. So we have here again an overview of the types of gene therapy techniques used in HCC treatment or the treatment of hepatocellular carcinoma. So we have here another diagram showing the strategies of in vivo gene therapy and the ex vivo gene therapy. In vivo gene therapy on the left starts with placing the therapeutic gene into the viral genome. The viral vector is then injected into the body via IV intravenous, IT intrathecal, IM intramuscular, or IP, intraparenchyma to transduce target cells. We also have the ex vivo gene therapy on the right. It starts with an extraction of the stem cells from the body. The virus with a therapeutic gene will transduce the stem cells outside of the patient. Once the viral vector has successfully transferred the gene to the cells are reintroduced into the body via IV, IT, or IM, where the stem cells can differentiate and the gene can be expressed. We also have here another diagram showing how the gene therapy works. So in this diagram, we have here gene therapy that uses a viral delivery system, and that virus is the adenovirus vector. So a new gene is inserted directly into the cell, and a carrier called a vector is genetically engineered to deliver the gene. An adenovirus introduces the DNA into the nucleus of the cell, but the DNA is not integrated into a chromosome. Gene therapy is currently aimed at treatment of diseases not responding to conventional drug therapy, such as inherited metabolic diseases, through either correction of a deficiency or of an abnormal metabolic reaction. It is also applied to induce the synthesis of a deficient protein in the host cells and the treatment of a variety of cancers, such as melanoma, renal carcinoma, lung cancer, cystic fibrosis, and others, and certain immunodeficiencies, such as HIV. Another drug source are the biopharmaceuticals. When we say biopharmaceuticals, these are also referred to as biological products or biologics, and they contain an active substance derived from or extracted from a biological system or living organism. They are often administered as an injection or infusion, and they are therapeutic agents produced through biotechnological means, but not by conventional laboratory chemical synthesis. The principle of biopharmaceuticals and the process of their development have origin from the advancement in the knowledge of molecular cell biology. The biopharmaceuticals, popularly called as designer proteins, are considered to be the promising therapeutic tools of the future. So we have here a slide showing what biopharmaceuticals are all about. So the biopharmaceuticals are also defined as a class of therapeutic agents produced by modern biotechnological techniques like recombinant DNA, protein engineering, and hybridoma technologies, and others. They are also defined as a protein or nucleic acid-based pharmaceutical substances used for therapeutic or diagnostic purpose, which is produced by modern biotechnological techniques. So these are some examples of biopharmaceuticals. So they include vaccines, blood or blood components, allergenics, somatic cells, gene therapies, tissues, recombinant therapeutic protein, and living cells. Other examples are hormones, particularly insulin. You also have the growth factors, the blood proteins, the clotting factors, enzymes, antibodies, and the stem cells. Specific examples of biopharmaceuticals include functional human peptides such as the hormones ADH, antidiuretic hormone, oxytocin, gonadotropin-releasing hormone, adrenocorticotropic hormone, thyroid-stimulating hormone and thyroid-releasing hormone, calcitonin, insulin, somatostatin, growth hormone, and others. We also have the enzymes or the proteins. So example of this are streptokinase, asparaginase, DNase, erythropoietin, interferon, and others. So these are the specific applications of biopharmaceuticals in health and medicine. First, the therapeutic proteins can be used for treatment of diseases. The vaccines can of course be used to prevent diseases. Protein or DNA-based diagnostics, regenerative medicine technologies, and for gene therapy. Of the approved biologics or biopharmaceuticals, their date of commercial production, their indication, and the company which manufactured the biologics. We have here the Homoline, human insulin. So this was first commercialized in the year 1982, and this is indicated for the treatment of diabetes. We also have the TPA, tissue plasminogen activator, and this was commercialized or produced in the year 1987, and this is indicated for myocardial infarction. We also have the Epogen. Epogen is biologics that was produced in the year 1989, and this is indicated for anemia. The Betaseron. Now, the Betaseron is commercialized in the year 1989 for multiple sclerosis. Lastly, we also have the Pulmozyme, an enzyme, specifically a DNase, that is indicated for cystic fibrosis. Through DNA recombinant technology, bacteria are induced to produce hormones like insulin and growth hormone and enzymes like L-asparaginase. Production of insulin is important not only for human medicine but also for veterinary medicine. It is said that insulin production is made possible through recombinant DNA technology. But how did scientists make insulin from recombinant DNA? Recombinant DNA is a technology scientists developed that made it possible to insert a human gene into the genetic material of a common bacterium. This recombinant microorganism could now produce the protein encoded by the human gene. Scientists build the human insulin gene in the laboratory. Then they remove a loop of bacterial DNA known as a plasmid, and they insert the human insulin gene into the plasmid. So we have here now a plasmid containing the human insulin gene. Researchers return the plasmid to the bacteria and put the recombinant bacteria in large fermentation tanks. There, the recombinant bacteria use the gene to begin producing the human insulin. Scientists harvest the insulin from the bacteria and purify the substance for use as a medicine for people.